like elementary.
I know.
Like the elementary.
Get the show on the road. I don't know where everybody disappears to, but oh well. Okay, so the PM people yesterday suggested that the lab exam be on, instead of Monday, on Thursday. No, not Thursday, but Wednesday, sorry about that. On Wednesday. So I said I had to consult with the AM people, and so people were here. In fact, AM, PM, how many of you next week want the lab exam on Wednesday? Raise your hand. This includes the PM people too. Everyone who wants it. Okay, if you want to, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. How many want it on Monday? Okay, all right, looks pretty unanimous. How many don't care? All right. So the lab exam is going to be on Wednesday, next Wednesday, okay, just before spring break. That means that on Monday, I will go lab by lab, talking about some of the general features to remind you what you've been over. Okay, I might have time to do that tomorrow. We'll see. Okay, so lab tomorrow. The only pre-lab that you have will be for exercise 10. That's all, exercise 10. Exercise 8 will be due at the beginning, okay, but not exercise 9. Okay, because exercise 9 we still have to do a earthworm dissection, so you'll be doing the earthworm dissection tomorrow, and I'll talk about that when we get there. And then on Monday you'll be doing the cockroach dissection, okay? And then we'll figure out what we do for the rest of the week. Okay, so where I left off, we're making the run through the protostomia, and this is the phylogeny thus far that we have gone through. Okay, mostly constant. I didn't fill in Tropozoa, but you should know Mollusca is up here, Platyhelminthes is up here, and Annelida is up here. And there are various subgroups, okay? You have to be aware of those. But with the Ecdysozoa, the main apomorphy there is that outside of the epidermis, they have a cuticle. Okay, so this is mostly collagen, except for in the Anthropoda, this, instead of just being collagen, it is chitin as well. Okay, and it has an articulated exoskeleton. That is part of their cuticle. All right, and therefore they have to grow by molding. And so within the Arthropoda, the biggest group of organisms altogether. Okay, the number of species in this group outnumbers all other forms of life, including bacteria. We put them all together in terms of numbers of described species. Trilobites, they were they're extinct. Just let you know that they were really abundant during the Paleozoic. They went extinct at the end of the Paleozoic, along with that very mass extinction at the end of the Permian. The other two major groups that are alive today, Chelicerata, arachnids, so this includes spiders, ticks, mites, and then what we're into right now is the mandibulae. Okay? And so mandibulates, mouth parts or mandibles. Okay, that's apomorphous and apomorphic for this group. They're derived from a different segment. The head in these groups are segments that have fused together, okay, into a large structure. But the mandibles, they're not of the same evolutionary origin as the Chelicerata and so forth. They're different. In fact, there's two pairs of mouth parts. There's mandibles and maxillae. I don't have it listed up here, but this is what the mandibles would look like. In an ant, they're jaw-like structures instead of fang-like structures. Fang-like structures are chelicerate. These have a sensory organ on the head called antennae. Chelicerates down here do not have antennae. And so this is for chemical sensory. Chemical sensory, what they taste and smell with. Okay, so this group includes centipedes, millipedes. A big group that used to be treated all on its own is crustaceans. This group is one of those, once again, paraphyletic groups. that I'm not going to divide it up, even though, because that's still in the infancy of how it's being figured out what it is. But it is paraphyletic, and then the biggest group of all the insects. Okay, insects and spiders are not the same thing. Okay, spiders belong way over here in the Glycerata, whereas insects are in the Mandibulata. Right? Okay, insect groups, they start showing up in about the Paleozoic. Okay, so the subgroups within the Mandibulata. One of them called Myriopode. And this includes centipedes and millipedes. Okay? Centipedes and millipedes belong to this particular group. And all I'm going to say as an anapomorphic for it is internal structure inside the head. It's actually called I'll just put up here, fancy tentorium. Okay, a tentorium is part of the exoskeleton that vaginates inside of the head and provides a lot of bracing structure internal to the head. Okay, a lot of the muscles that operate the mouth pieces happen to hook onto the tentorium. All of these have mandibulates have a tentorium. Okay, this is inside their head. But this here, instead of fancy, let's be a little bit more scientific. Okay, complex. Okay, so it's a little bit different than the tentorium that you find inside the head of other arthropods. And here I just want to say this is actually a genetic instead of molecular, instead of DNA sequences. It has a lot to do with the expression of a particular gene called ultrabithorax. Ultrabithorax is one of several Hox genes. These Hox genes in animals, they're expressed in early embryology. And their expression determines what is going to be the head, what is going to be the tail, what is going to be left, right, what is going to be top, bottom, and so forth. Yes?
Is the complex tentorium the same as the unique internal head structure?
Yeah. I've just given you a much better name to make it shorter. Instead of having to say unique internal head structure, that's a little bit long. Just say the complex tentorium, shorter. And also it's a fancy word, right? We like fancy words. Okay, but yeah, so ultrabithorax is one of those genes. And so all I'm going to say here, but I'm not going to explain what strong antisense expression happens to be, simply that all animals have it, okay, this ultrabithorax gene. But it has a special type of expression in this group, okay, that's different from other groups of animals, right? And that's all that we have to say for this particular Myriopode. The two groups that belong here, Myriopode or millipedes. And Biol-101B, which are the centipedes. Okay, so first of all, Biol-101B. Now the tagma, remember what tagma happened to be? Tagma is singular. That's a special body region. It consists of many modified segments. For example, in the head or the cephalothorax, there's many segments fused together, and so you really can't see the segmentation there very well. But so in down here in the glycerates, the two major tagmata, okay, tagmata is plural, tagmata is singular, is the cephalothorax and the abdomen. Cephalothorax and abdomen. But in the centipedes and millipedes, these regions are a little different. Instead, there is a head and a trunk. Okay, the trunk carries all of the legs. Okay, so there's a pair of legs for every abdominal segment. The head is actually 6 fused segments. And I'll never ask you about how many segments are in the head. I just want you to know that there are several anterior segments that have fused together to form the head. All right? So body consists of a head and trunk. Trunk has all the walking legs. Now for the diplodocus, they appear to have 2 pairs of legs per segment. Okay? Now this millipede, milli meaning 1,000, they don't have 1,000 legs. Okay, nowhere close to that. But some of them can have up to 100 pair of legs. It differs from species to species, genus to genus. Now, really, ancestrally in the Arthropodum, we believe that every segment had 1 pair of appendages. So how did they end up with 2 pairs on a segment? We believe that these are 2 segments that are fused together. Okay, in fact, in the embryo of these things, those are separate segments. And during embryological development, they fuse together, every other one of them. And that's exactly what this sort of di, meaning 2, and ploda is referring to segments. So these are 2 fused segments together, all right? And it makes it appear like they have 2 pair of legs per segment. All of these are herbivorous. However, some of them will produce at the bases of their legs poison glands, so that they secrete poison. Sometimes when they're attacked by a bird, a predator, they will roll up into sort of a spiral structure where they have their underside tucked in, and they will secrete this poison, toxic, at the base of the legs, and it gets the bird sick, and they'll leave it alone a lot of times. Okay, but they're all herbivorous. Or they feed on organic decay. The Calopoda, these are the centipedes, and they're all predators. And they're all poisonous. Okay, just like the millipedes, in fact, that's true of all Myriapoda, is that they consist of a head and a trunk. The trunk has all the walking legs on it, right? So, major anthropomorphy here for the Calopoda. Okay, now they only have 1 pair of legs per segment, but that's ancestral. Is that the first pair of legs, it's hard to see it right here, but the first pair of legs on the trunk are not used for walking. They have been modified into a poison gland or poison fangs. They're real hard, and they are associated with poison glands. They will inject this poison, first of all, stab their prey with these front legs, and then secrete poison into the animal. Some of these get to be pretty big, especially in South America. There's a genus called Centroides that they can be over 1 meter long. Can be really quite big. But all of them have poison glands, and all of them are predators. Predatory, okay? And then another clade of Mandibulata. Put it a little bit sideways here. Pancrustacea. All right. Now this clade has been put together simply to make a monophyletic group, okay? Because there's 2 groups of organisms here: insects, crustaceans. I already said that the crustaceans are paraphyletic, okay? Just like the bryophytes are paraphyletic, and a few other things that we've come across being paraphyletic. But because we now understand that the evolution of insects are just highly modified crustaceans. They're more closely related to one group of crustaceans than what they are to other groups of crustaceans. They arose out of the crustacean, which would mean that this taxon that we call Crustacea, that you went through high school with, is paraphyletic if you leave out the insects, okay? So to make a monophyletic taxon, we have Pancrustacea, which is all the crustacean groups plus insects. They all belong in here. And there's a lot of evidence here besides DNA evidence about the modifier. One of it has to do with the structure of their compound eyes. Now, arthropods have simple eyes. Simple eye is just chitinous lens that's over a section that has light sensing neurons that then take the message to the brain. This type of simple eye cannot focus. All it can see is movement, light, dark, shades of light, dark. But other than that, it doesn't see anything, all right? Saw that in spiders, okay? That's called a simple eye. But both in the myriapods and in pancrustaceans, they have a compound eye. A compound eye is made up of lots of little rod-like structures that are called ommatidia. Hundreds of them next to each other. Once again, this ommatidium cannot focus. It does have a lens up here, and on the sides, or mostly down in this area, there's light sensing neurons. But however, these light sensing neurons can only pick up light that's coming directly down. Okay, light that's coming from the side gets absorbed by these pigment cells on the side. So it can only see, each little ommatidium can only see a pinpoint of light that's coming directly into it. But it's made up of hundreds of ommatidium that are on eyes that are curved. And so what they see, and they can see not just only in the little plane out here, but sometimes all the way around behind them, is a mosaic of light, little pins. If you take a picture and then zoom in, zoom in, zoom in, you see that the picture is made up of all these little colored dots. But when you shrink that down to the point of our eyesight, it looks like a very, very clear image. And that's how they see. They do see images, but it's not focused, okay? It's just due to pinpoints of light, all right? So these are compound eyes, and the compound eyes that are in the myriapods is somewhat limited in the number of ommatidium. Predators in the insecta, they usually have sometimes up to thousands of ommatidium, and as a consequence, they have pretty good eyesight, right? This is why it's hard for you to slap a fly sneaking up on it, because it can see behind you, and over here, and over there, and up there. They can see all around. Okay, so this lens up here, so that's the ommatidium. But what is the apomorphy that's found in all pancrustaceans is that there's 4 cells up here at the top of the ommatidium that secrete the crystalline cone. This is the lens. And so all of them have that pattern of 4 crystalline cone cells up at the top of the ommatidium. Okay, so crustacea, I'm not going to put it up here. Well, yeah, I will. It's paraphyletic. I'll put it in quotations. Okay, this group included things like crabs, lobsters, krill, shrimp, crayfish. It was very copepods and a few other things you don't come across. Isopods. Anyone know what an isopod is? We have them here on our, if you turn over logs, you see one terrestrial isopod that are often called roly-polies. Okay, those are crustaceans, right? And barnacles, okay? All these happen to be crustaceans. And strikeout probably, it is paraphyletic, and there's good evidence of that now. And some of these aquatic ones will have, a variable number of legs, and they have 2 pairs of antennae a lot of times. Not all of them, but some of them have 2 pairs of antennae. And the second pair of antennae is derived from lay-like structures. They do have compound eyes. Those that are aquatic, they will breathe by means of gills. And these gills are structurally quite different than the gills of fish or vertebrates. Okay. As I mentioned before, this word gill and lungs, throughout the animal world, these words are by analogy. Okay, they're not, doesn't indicate any evolutionary relationships. Those that live in the ocean, a lot of times in addition to their jointed exoskeleton consisting of collagen and chitin, but they may also have calcium carbonate deposited in it, which makes their shell, especially in crabs and lobsters, crayfish, makes the shell very, very hard. Terrestrial ones don't have that calcium carbonate. It's a very widespread group, but out of it comes one monophyletic group, namely the insecta, or Hexobida. You can use either word. So I'm just going to draw it out of the crustaceans. And this is equal to insecta. Okay. All right. Yes.
Is it out of Pancrustacea or Crustacea?
No, Pancrustacea is insects plus all crustaceans. Okay. So that clade.
Okay.
All right. Which is monophyletic. But it is paraphyletic. Crustacea is paraphyletic because it is all pancrustaceans except hexapods. And these are more closely related to some other group here, and they already have a pretty good idea as to what crustaceans they're most closely related to, but we're not going into that. All right, so Crustacea by itself is paraphyletic. Okay, the tagma that we have in tagmata in this group is that there's three body regions. A head, a thorax. The thorax has the walking for locomotion. The thorax will consist of three segments in the thorax, and each segment has a pair of legs. So they have three pairs of legs, right? And some of them on the mesothorax, that is the segment in the middle, the metathorax at the end, will have a pair of wings as well, but not all of them, maybe just some of them. So this here is for all of the hexapods, and as a consequence, they also have three pairs of legs. And so I'm going to go ahead and put that down as an apomorphy. And within Hexopoda, we have several groups down here that do not have wings. One group that called Pterygoda, they have evolved wings. Okay, so down here, silverfish. Something called Columbella. And a few others that are primitive groups. You don't need to know the Columbella, but things like silverfish and a few other primitive insects, they have no wings in their ancestry. They evolved before pterygotes evolved wings. So this is only, so wings is only an apomorphy for the pterygoda, not for anything else. Okay, so this is a very large group, very successful, and as you go through all of it, you'll find here and there that there are ones that don't have wings. Okay, now for silverfish and springtails, okay, Columbella, the common name for it is springtails. These are real small little insects. If you turn over leaves, you'll see hundreds of them, and they jump around and everything. They're ancestrally wingless. That means that they did not come from an ancestry with wings. But there's other things, such as, say, Okay, I'll come back here. Other groups, such as fleas. Fleas have wings? No. Fleas don't have wings. What about ants? No. Termites? No. For the most part, they don't have wings. But we know that they came from an ancestry with wings. They just secondarily lost it. How do we know? Okay, it has a lot to do with the structure of the thorax. Okay, so here in the silverfish, as in also centipedes and millipedes, each segment consists of a plate on top. It's called a tergum. This calls it a notum. I prefer the word tergum. That's a dorsal plate. And then a ventral plate. Okay, a tergum and a sternum. Tergum on top, sternum on the bottom. And around the legs, they have a few hard plates, small plates. They're called sclerites. Okay, these things brace the leg, and then between the sclerites, tergum, sternum, is a loose membrane. It's just called an intermembrane structure. Just call it membrane. It is flexible. Now, as wings were evolved, this had to change because the wings, there's no muscles usually directly attached to the wings, except for in a few groups. But as the muscles contract, then it would just pull this all together, and we wouldn't have any sort of movement of the wings. So these sclerites had to expand so that they would fill in, all this membrane would disappear, so that the edge, the side of the thorax would become very, very solid and well enforced. Okay, so here's where the legs are. Those sclerites have expanded. The tergum is up here, sternum down here, so that this thorax is a very solid box without any membranes between the sclerites so that it doesn't collapse. And then the wings are up here, okay? So as these, what powers most of the wings are muscles that go from the tergum down to the sternum. They're called flight muscles, dorsal-ventral flight muscles. And the movement of those muscles does sort of deform this box enough so that it moves the wings in very specific ways. Okay, these critters, ants, fleas, lice, they don't have a thoracic segments like this that has all this small sclerites and a lot of membrane. They have this type of reinforcement. And so that's indicates that they came from an ancestry that had wings, and they've subsequently lost wings. Incidentally, some ants do have wings. These are reproductives, not the workers, but the reproductives. The males and females that will reproduce the colony, they do have wings. Fleas never have wings. Some lice never have, well, yeah, they don't have wings either, okay? But nonetheless, this thorax like this, highly reinforced thorax, is an indication that they came from a winged ancestor. Okay, for this example, here's a flea. Look at that area. You see that all those sclerites are expanded, and there's no membranous material around it. The only way that that could come about is with an ancestry coming from something that had wings. Okay, now I said that the arthropods were more numerous than any other group of organisms. Well, the insects is the main reason, because there's more species of insects than anything else. They're a very successful group. Okay, this is just giving you some examples of insects. It's the beetle, it's the silverfish, this is the midge, and praying mantis, ants, but they're very, very varied. And some of them can be real big, but the vast majority of insects are very, very small. And you find them everywhere. Okay, now there's three different patterns of in insects of post-embryonic development. Now, what do I mean by post-embryonic development? Embryonic development is inside the egg. Okay, that's where the embryo develops. Then once it hatches from the egg, these insects will go through several different stages of development, and that's what I'm talking about. Between the time they hatch from an egg and reach adulthood. Okay, three different patterns. One of them is called ametabolous, and this is what you find in springtails, silverfish. Ametabolous. And actually, it's found in all other arthropods, but we don't use this word for characterizing the post-embryonic development of other arthropods, simply because they're all that way. Okay, they're all ametabolous. But we have three patterns only here in the hexapoda, in the insects. So ametabolous, when they hatch, they look like the adult, and they will molt as they grow, and they continue to molt after they reach sexual maturity. Okay, this is unusual because in the other two groups of insects, once they reach sexual maturity, they no longer molt. That's the end of the story. Okay, so these continue to molt afterwards. And furthermore, the young and the adults, they eat the same food supply. So they compete with each other. Second group is called hemimetabolous. And I'm not putting these up in any phylogeny, okay? These are just only developmental patterns. Is that with hemimetabolous post-embryonic development, is that the immature, once they hatch out of the egg, they resemble the adult in certain ways, but they lack wings, okay? So these are only the next two groups that I have here, hemimetabolous, and the next group I'll talk about, holometabolous. They're pterygoda, okay? They belong to that group. Ancestrally winged, all right? So these do not have wings. They will have external wing pads that inside are embryonic tissues that will develop into wings when they make their last molt. Their last molt is into the adult stage. So they are now sexually mature and they have wings. Okay, so if you find an insect running around that doesn't have wings, or let's put it this way, you find an insect running around with wings, this would be the easiest way to say it, it's an adult. Only the adults have wings, okay? If you find an insect that doesn't have wings, it could be an immature, or it could be the adult of one of those species that has lost its wings, which has happened many times in the insects. Yes?
So you already said that some ants have wings?
Yes, the reproductives, males and queens.
Does it have any function?
Yeah, they fly.
Oh, they do.
Yeah.
Thank you.
Yeah, so what's happened with, okay, an ant colony? Ants, incidentally, are glorified wasps. They belong to the same group. They're very closely related to wasps. And when the colony gets big, these are social insects. They've evolved a society which most of the members are females. So all these ants that soldiers that protect the den, do all the work, those are all females. The only time they produce males is when the colony gets too big and needs to split it. And so they will produce reproductives, males, females, that have wings. They fly away and far away and start a new den. Okay. Termites, too, they will have winged reproductives. But termite dens, the workers are both males and females. It's not like in ants.
Sorry, going back to the last slide for ametabolous organisms, how do mature insects recognize other mature insects as opposed to like immature ones? If they
Okay, how can you tell them apart?
Yeah.
Okay, well, usually have to pick them up, turn them over, and look at their genitalia.
okay? Because the young will not have them. They won't be sexually mature, okay?
No, I meant like how do members of those insects recognize other members?
Oh, okay. Now that's by smell.
Ah.
A lot of times, because a lot of the insects produce chemicals that are called pheromones. And the pheromones, the composition of chemicals that are in that perfume is pretty species specific. You know, so it's, and in fact, to catch certain pest species out here to see if they're abundant, the county will put out little traps that they hang up in trees. And usually that has a pheromone in it that will attract a target species that they want to find out if it's here in this area, okay? But yeah, it's primarily their pheromones, and they can tell each other apart. Okay, so this is the hemimetabolus, and so the immatures are called nymphs, okay? So they're not larvae, they're called nymphs, all right? And so insects that are of the hemimetabolus group, grasshoppers, crickets, cockroaches, aphids, praying mantises, walking sticks, and a whole group that are called the true bugs, hemipterans. And so all those are hemimetabolous. The last one, more complex, is holometabolous. And this group of insects, I'm not going to put it up here, it is monophyletic, okay? Definitely monophyletic, because this hemimetabolous development is quite unique. And here, usually the immatures, when they hatch out of the egg, is they look completely, for the most part, they look completely different from the adults. For example, flies, they're worm-like, called maggots. Okay, even with beetles, a lot of the immatures are grubs, and they don't look quite like the adults. So these will go through molt, and here the immatures, since they look so different from the adults, are called larvae. Okay, so in the hemimetabolous, the immatures are called nymphs. These are called larvae. Okay, larvae. Then the larval stage is a feeding stage. Feeding stage, growth stage. Usually there's a limited number of molts, sometimes no more than 6 different molts. One of the last molts will transform the last larva stage into a resting stage called a pupa. Sometimes this is as the case it is in flies, is that the pupa is developed from the last larval skin. But in other things like moths and butterflies, they spin a silk cocoon, and pupa is inside it. Now this is metabolically very, very active. Even though they're not feeding, what happens here is that larval tissues are completely being broken down and adult tissues formed. Incidentally, the immatures, the larvae here, do not have external wing pads. Okay, the hemimetabolous, they have external wing pads, embryonic wings. These here are internal. They do have wing pads, but they're inside the body. And when they go through the pupal stage, these wing pads evert outside, and they develop into wings. Then, once it ecloses from the, they don't hatch from the pupa. It's called eclosion. Put that word up here. Okay, when they eclose from the pupa, they'll emerge as an adult fly with wings. And this is the holometabolous. Now, one of the evolutionary advantages of this is that the larvae don't compete with the adults for food. They eat totally different stuff, usually, yes.
So would a caterpillar be a larva?
Caterpillar is a larva. And most of the caterpillars that you find running around, when they transform into adults, will be moths. Okay. There's some that are more colorful that will become butterflies. Okay, so butterflies and moths, they're all belong to the same clade. Okay, so this has mosquitoes, beetles, moths, butterflies, lacewings, and wasps, bees, ants. They all belong to this holometabolous group. Okay. In fact, the number of beetles, there's about described 500 million species of beetles. There are not million, 500,000 species of beetles, real big group. Okay, so this is an important group because it has a big impact on humans in terms of being disease vectors. So in this particular use of this word vector, it means an animal, an agency that transfers a disease agent, okay? They don't cause the disease. The disease is caused by a microorganism of some sort. But many of these are blood-feeding insects, for example, malaria and African sleeping sickness. They are spread by the bite of a female mosquito. Okay, in the case of malaria, it's only Anopheles mosquitoes. That's one genus of mosquitoes. Other genera of mosquitoes, such as Culex, Culicida, and others, they will not carry malaria. So it's sort of restricted, and not even all species of Anopheles mosquitoes will spread it. African sleeping sickness, spread by the tsetse fly. Tsetse fly is still a two-winged fly related to mosquitoes, but it's more similar to a house fly. Bubonic plague. This is not a big deal in the United States anymore, except for every year there's a few cases of bubonic plague. But during the Middle Ages, it was a big deal. So there was a wave of bubonic plague during the Middle Ages that probably reduced Europe's population by about 1/3. Okay, quite a few people died from it. Now it is easy to cure with antibiotics if it's caught early enough. It's spread by the bite of fleas that would normally live on rats. Filarial worms, I already told you about nematodes being spread by the bite of a mosquito. These filarial worms get in the body and they cause the legs to swell up as the worms proliferate. I showed that picture before, elephantiasis. That's spread by mosquitoes. Yellow fever is Aedes aegypti mosquitoes, and this is caused by a virus. So this is a virus, yellow fever, dengue fever. These are spread by mosquitoes, bites, but it's a viral disease. And many of our crops, there's all sorts of viral diseases that plant-eating insects will spread. So this is a pretty important group. Furthermore, some of these are getting to attack our crops and destroy them, our food supply. Some of them get into stored products and cause a lot of waste there. And of course, termites will attack our homes. They're made out of wood. And so these cause billions of dollars of economic damage each year. But some of them are good, okay? There's a lot of insects that are predators, and they are eating other insects, including pests. Now, defining what a pest is, it's sort of from our own standpoint. Anything that we call a pest is simply because it is causing economic or health damage to a human being. Okay, now this particular thing about predators eating some of these pests led to a different type of approach to controlling agricultural pests called biological control. This was developed during the 1950s, or even earlier than that, at UC Berkeley. UC Berkeley was really big on biological control. Unfortunately, most of the entomology department at UC Berkeley has disappeared. In fact, I was probably one of the last PhD students to ever get a degree in the department, and now the department's gone. They moved a lot of it to Davis. But what biological control is it's instead of using pesticides extensively, which are rather promiscuous, they will kill non-target organisms a lot of times. They're usually broad spectrum and cause a lot of damage. The whole idea of biological control was to find some enemy of the pest that you could manipulate into controlling the pest. Now, how do these things become pests? Usually they have been introduced. They're not native to the country in which they are causing all the damage. Wherever they came from, they evolved in an ecosystem where there were diseases that controlled them, there were predators that controlled them. So once they've been moved out of that habitat into a new place where those predators and diseases are not located, then there's no check on their population and they become pests, okay? So a good story here, successful biological control, had to do with something called the Cottony-Cushing scale here in California, okay? During the late 1800s, early 1900s. In Southern California particularly, they grew a lot of citrus, oranges, lemons, and other things. And this is a Cottony-Cushing scale is an insect that just devastated the citrus industry. It is not native to the United States. So they sent out several entomologists from here in California to try to find out where this thing originated in the world. They finally found it in Australia. Okay, in Australia, it's not causing any problems whatsoever. And they did studies to try to find out what is it that's controlling the populations in Australia. And they found this beetle, which looks like a ladybug beetle, okay, the dahlia beetle, that they keeping these under control. So they brought the dahlia beetle back to California, did experiments, hoping that it wouldn't cause any damage to the existing environment, and they mass released it in citrus groves, and it did control this. Now the whole idea was not to completely eliminate this, because if you eliminate the Cottony-Cushing scale, this has nothing else to eat, because it only feeds on that, and it would disappear. This would come back, and we'd have problems again. So the whole idea was to maintain both of these in low levels, so below economic damage. So we have to tolerate a little bit of economic damage, but not as much as before. And so this was a very successful story. This is what the Cottony-Cushing scale looks like, and this is what the dahlia beetle looks like. Okay, and they do occur around here. In fact, last summer, I have a orange tree. I did find some of these things on it, but I took a different approach. I just picked them off. Okay. Now another biological control that is being practiced quite a bit and experimented at Davis primarily and Riverside is the use of parasitoid wasps. Okay, so wasps, these particular wasps are incredibly small. You usually, when you think of a wasp, you think of something pretty big, but these are like little tiny midges. But what they do is they lay their eggs inside other insects. So they have a needle-like structure called an ovipositor. They'll land on, say, a caterpillar, put the ovipositor in, and lay the egg inside of the caterpillar. When the egg hatches, you have these grub-like wasp babies that eat out the inside, eventually killing the caterpillar. They usually choose very specific places to eat to keep the caterpillar alive long enough for them to reach adulthood, or at least pupate, and then they leave. So, how many of you have ever seen this movie called Aliens? Okay, at least a few of you, okay? And let's see, Hollywood, these things beat Hollywood to that idea. They evolved it millions of years ago. Okay, so this is a parasitoid wasp, another parasitoid wasp. This is an aphid. This is laying its egg inside that aphid, okay? This is a moth larva. This is something called an ichneumon, and it's laying its egg inside that moth. Okay, and then furthermore, we get food from insects as well. Honey from bees, it's a silk is produced by a tobacco moth called Manduga. And it's, a lot of places we get over our fear or our queasiness of eating insects. They're a good supply of protein. Some places in the world, they do cook these insects, and it's worthwhile. Pollination, you can't, they're major pollinators for lots of plants out here, including crop plants. And right now, a lot of the honeybee populations we have, commercial honeybees, they're catching diseases, dying down, and so we're really concerned about pollination because we depend on them. Okay, so that is protostome. Any questions? Because now the next part is to go into heterostomia. Okay, any questions? Yes?
For the holometabolites, what is their, molting pattern?
Was there what?
Molting, like how do they molt?
Okay, usually they will split along the dorsal side. There's an area that is very, very thin. And when they're ready to molt, the epidermis, because the epidermis is under all this, has glands that secrete digestive material that's in between the epidermis and the cuticle, and it breaks down some of the cuticle. Okay, they can't break it all down. And then along the dorsal side, there's a thin area. Once this juice has broken down some of the cuticle, they puff themselves up by taking in a bunch of air or water, stretch, and it splits. Then they have to pull themselves out, and they leave behind the exuvium, okay, which is the old exoskeleton. Okay, sometimes you can find these things. If you go around, it looks like it's just a thin piece of insect, dead, but it has a split along the top. It's just shed skin.
So is that different from ecdysis?
That's it. This is ecdysis.
Oh.
It's the same thing. So all these in the ecdysozoa, including nematodes, they all molt, okay? And very much the same way. Okay, they have to, first of all, break down some of the cuticle that's closest to the skin, but they don't break down all of it. Then they have to puff themselves up with air to create, or water, to create pressure, to split it open. Then they have to crawl out and leave behind the old skin, the old cuticle, and then puff themselves up and secrete a new cuticle. And that's the same here. Yes?
After the caterpillar dies, does the parasitoid wasp just find a new host to?
No, say what?
After the caterpillar dies, does the parasitoid wasp just find a new host to?
Yeah, they will. Yeah, so usually when the caterpillar dies, it's about the time that what will break out of here will be the grubs, and then they form a pupa, usually in the dirt, and then from the pupa, the adult will emerge, and then the adults will mate and find new hosts to lay their eggs in. Yeah. Okay, yes.
Can you give an example for mutabolous insects?
An metabolist?
Yeah.
Silverfish.
Silverfish.
Silverfish. Because silverfish, and there's others small, there's a few groups of ancestrally wingless insects. The ones that they're real rare, except for bristletales and silverfish and springtails. And springtails, there's about 5 different families of springtails. They're usually very, very small, white things that you find in leaf litter. Okay, but they're all ametabolous.
So would they, so they're not, so ametabolous insects are not a part of
Not pterygoda.
Yeah.
Yeah. They're insects, they're part of Insecta, but within Insecta, there's a larger taxon called pterygoda. That is monophyletic. That is monophyletic. Okay? All right, so now we're going to go to the last part of the animals, namely the deuterostomes. And there's only two major groups here and down here that we'll discuss, but one of them is pretty big.
Sorry, I just have a quick question about the heavy metabolist types of organisms. Do they also have competition between like the children and the?
Okay, question here is with the hemimetabolists, is there competition for food resources between adults and the nymphs? Yes, they do eat the same thing. The holometabolists are the ones that really escape that. Yeah. Okay, yes?
I had a quick question on Diplophoda.
Yeah, Diplophoda.
You said that for the apomorphies, it is believed that the two segments are fused together.
Yes.
Could you just clarify?
Okay, in all of the arthropods, each ancestrally, we believe that these were like earthworms. They had segments. In fact, we still can see the segmentation, especially in the abdomen. But primitively, each segment had a pair of appendages. Not necessarily legs, they might function as something else, but they had a pair of legs per segment. Okay, so in the diplopods, they have two pairs of legs per segment. So how did that come about? Well, this is due to two segments that were independently separate from each other, fusing together, each of which had a pair of legs. Now, we can even see this in the embryo, okay, because in the embryo, those few segments are separate from each other. And as the embryo goes along, then they fuse together. Okay, this is where they get two pairs of legs. Okay, deuterostomes, the last flow through here. One major group is the echinoderms. Okay, so I'm going, if you want to take a photo of this, you better do it now. Okay. I'll even let you come up. I'll pause here for a little bit. You can come up and take that. Or if you don't like walking, you can stay in your seat and just zoom. Okay, because this is going to disappear.
No.
What?
It's okay.
You're still trying to get it?
No, I got it.
You got it? Okay. Deuterostomes, chemodermata. Okay, the echinoderms. These are starfish, sea urchins, sea cucumbers, and other types of things related to that. And they're all marine. You don't have any freshwater ones. You don't have anything that are terrestrial. This is a sea urchin, this is sea star, sea urchin, sea cucumber. Okay, and they're all marine. And what apomorphy for this particular group is that they are have radiosymmetry. Don't they belong to the radiata? No. They have all of the other apomorphies that I have mentioned all through this tree that I happen to put up here. This is a different type of radiosymmetry. It's pentaradial. So unlike in the Cnidaria where you can cut through the aboral axes, oral axes in many different slices to have mirror images. Here, there's just only 5 different planes of mirror images. Okay, and furthermore, the larvae, they're called larvae, the immatures aren't adults, they are fully biradial. They have all of the characteristics of any sort of bilaterian animal. So this is a derived character, okay, it's secondary. They still have a complete digestive system, because when we looked at the radiata, they had a gastrovascular cavity. Okay, just one opening, one opening. Here, they still maintain a mouth and an anus, and they have a fairly complex stomach. In fact, there's two stomachs, one of which they can evert outside its mouth. Talk about that a little bit later, and so forth. So they are, this is a secondary condition. Bilateral condition is still in the larva, immatures. They have evolved something that is called a water vascular system. This is how they both feed. It serves for feeding, it serves for motion, locomotion, and also for respiration, gas exchange. And so part of this water vascular system, which I'll show here in a moment, involves hundreds and hundreds of flexible little tubes with suction cups on the end of them that are moved and manipulated not just by muscle but by water pressure, that are called tube feet. Okay. Since these adults, the adults are have radiosymmetry, they have an oral side and an aboral side. The oral side is where the mouth is. The aboral side is where the anus is. Okay, it's opposite the mouth. Okay. So this water vascular system, okay, some other characteristics here is that they are dioecious. They're separate males and females. And you really can't, in a starfish, you can't just look at them and be able to tell which one is male and female because they look very much alike. The differences in the testes and ovaries, which they have a pair of them in each one of these 5 arms. Okay? Like down here. But fertilization is external. Okay? Obviously, a male and a female would have to be in the same vicinity because if they're far away, releasing the sperm and eggs into the water, they will never meet. Okay? So they still get together and release the eggs and sperm. So this water vascular system, pretty complex tube system. On the aboral side, there is a sieve-like opening, and in lab you'll be able to see this, that is called a madreporite. And it's right a little bit off to one side from the anus. Okay, the anus is right in the middle, but the madreporite is on the side. This is where water is drawn in through the madreporite down a flexible, short stone canal. Okay? And so the stone canal is just taking water from the madreporite to another canal that is rather hard because it's made out of calcium phosphate and calcium carbonate. But anyway, it's a circular canal that goes all around like a ring in the middle, and that's called the ring canal. And then from the ring canal going down each arm is a radial canal. And along the sides of the radial canal, there are these structures that, if you looked at them up close, have sort of a stem like this, and they end up in sort of a little sucker-like structure down here. These are tube feet. And there will be hundreds of these tube feet. And they are for locomotion, and also they use these to capture some prey as well. All right? As far as the internal anatomy, it has 2 different stomachs. One of them is called a pyloric stomach, and the other one is called a cardiac stomach. So from the mouth, okay, actually this is the aboral side, because there's the madreporite right there. The mouth is on the other side, but the cardiac stomach is the first stomach. And this is evertable. What starfish eat, they eat clams. Okay? Clams don't move very fast. Neither do starfish. Okay, but what the starfish will do is they'll come up on a clam and take 2 of its arms, or maybe more, attach on each side, and all these tube feet attach, and it has muscles. It'll pull the shell open just a little bit, okay? And then its cardiac stomach, it'll evert out its mouth and stick it into the mantle cavity of the clam, where that releases digestive juices, okay? Eventually, those digestive juices weaken the clam, break it down, and soon the clam, or the starfish can open it up all the way and finish it off, okay? Clam fishers do not like these things, but unfortunately, clam fishers do not know much about the biology of the starfish. Whenever they catch clams with starfish in them, they would usually take a machete, cut it in several pieces, throw it back into the ocean. Well, these guys have wonderful powers of regeneration. Each one of those pieces, as long as it contained part of the central disc, would rejuvenate into a full starfish. And so instead of killing the starfish, they made 5 or 6 more. Okay, so it's good to know the biology of things before you mess with it. Okay, that's all I'm going to say about echinoderms. The rest of it, we're going to spend our time chordata, the group that we belong to, and that we're most familiar with. I'm going to put in one little group here. I'm not going to study it too much, but I want to mention it. Something called hemichordata. And I have a reason for putting that there. Talk about it here in a moment. Chordates. Okay, there's a lot of apomorphies, semapomorphies for this group. So I'm going to be mentioning quite a few. And as on any quiz or exam, of course, I hardly ever ask for more than 2 or 3. Okay, so you can choose which semapomorphy you want most. Okay, one thing that all chordates have is a cartilaginous, usually it's made of cartilage, flexible rod that is located between the gut, the intestine, okay, between the gut and the nerve cord. Okay, it has a dorsal nerve cord, and this is called a notochord. Okay, it is for, it's sort of a skeletal support for muscles. In the most basal groups of chordates, these muscles are arranged in segments. The earliest chordates were segmented. Okay, segmentation, you see quite a bit. They have a dorsal hollow nerve cord. Now, up to now, I've been mentioning nerve cords. Where have they been? Where are they located? Nematodes, I mentioned nerve cords. Should have done that in the arthropods. They're ventral. All these animals that I've mentioned have ventral nerve cords going down the bottom side of the body. Here, it's the other way around, dorsal. Okay, that's pretty unique. Not only that, but it's hollow. Okay, there's a fluid-filled space on the inside of the nerve cord, whereas the nerves in most other animals are pretty solid. Okay, the neurons are very, very close together. So this is somewhat unique. And they have a muscular post-anal tail. Okay, all the other animals that I've been talking about up to now, earthworms, insects, spiders, ticks, mites, nematodes, the anus is right at the very tip. Okay, but here the anus is forward, and so it has a tail that proceeds posterior to the anal opening. Okay, this is unique to chordates as well. Okay. And your textbook lists this as an apomorphic. It is not strictly an apomorphic for chordates. So I don't have stars up here because it's actually an apomorphy for a larger group, something called hemichordata plus chordata. And we're not going to look at hemichordates, just want you to know that they exist, but they also have pharyngeal slits. Now, what is the pharynx? We have the mouth, and right behind the mouth is always an area. Even in our own anatomy, we refer to this area here as the pharynx. Okay, but in early chordates, as well as in hemichordates, this pharynx has all these slits. these were filter feeders. Filter feeders. And the way in which filter feeding worked is they would take in water that has organic material into the mouth. It would get into this basket pharynx, and the water would move through these slits to the outside and exit the body. As that water goes through these pharyngeal slits, then there's mucus there that captures these fine particles. And then the mucus moves on down into the stomach area and into the intestine was digested, right? Now, in more advanced chordates, these pharyngeal slits have also developed into gas exchange structures that we call gills, okay? Gills come from this. Incidentally, human embryos will have those. They will eventually disappear though, okay? Because it's something that's in the early foundation of the embryology of chordates, and it's easier to keep it and change it later on in development than it is to do away with it, okay? Furthermore, some of these anterior slits will evolve into something else I'll talk about later. All right, so the pharynx actually happens in the hemichordates. Your book lists it as a apomorphy, but it's really something broader. As I said, this is originally for filter feeding, but later developed into a gas exchange organ as well. Gills. I think I gave you that whole statement, didn't I? Okay. Now, I'm going to give you some things that the book, your textbook, does not mention that also apomorphies, is an endostyle or thyroid. This is in the pharyngeal region, and it's a special type of gland that is involved in handling iodine, involved with iodine absorption. We don't have an endostyle. Endostyle eventually will evolve into a thyroid. Our thyroid is up here in the pharyngeal area, and we produce an iodine-containing hormone in the thyroid gland. We'll talk more about it a little bit later, called thyroxine. And one other that also your book does not mention. Chordates are sort of inside-out animals compared to everyone else. And I'm calling this dorsal and ventral inversion. And this has to do with certain morphogenic gradients that are inverted. I couldn't find a good figure, so I made my own. This here is in the hemichordates, and because they're the ones that have a pharynx. This is in the pharyngeal region, okay? But the slits, pharyngeal slits, are on the dorsal side of the animal, okay? Whereas in chordates, those with regard to the pharynx, the pharyngeal slits are ventrally located, not going up like this, but going down. We turned upside down. Now, these gradients for ventral and dorsal, they involve in the early embryo gradients of two different chemical compounds. Okay, these are used as developmental signals. One of them is called chordin, the other one is called bone morphogenic protein. So proteins, but it's in all other animals, it is those, the bone morphogenic protein is produced by cells that are on the dorsal side of the animal, whereas chordin is produced by cells that are on the ventral side. And as these diffuse away from where they're produced, then their concentrations decrease. And it is this gradient between these two that determine whether structures turn into dorsal structures, ventral structures, or somewhere in between. And so they set the dorsal ventral gradient during development. There's a lot of chemical signals that are released by cells that determine what part of the body develops into what, okay? Because these will enter the cells, bind to certain parts of the DNA, and turn on certain genes, okay? In chordates, the gradients are just the other way around, okay? They're turned upside down. We are upside down animals. And this is also sort of relates to where our nerve cord is. It's also in the wrong place when compared to all other animals. All other animals have ventral nerve cords. We have dorsal nerve cords, okay? And so this is what I'm referring to, is dorsal ventral inversion, okay? All right, so you won't find that in the book. Coordinate phylogeny, this is according to your textbook. We're down here, and this is where we're going, okay? All the way up to where we belong. And I'll sort of simplify this a little bit. Okay, so within the chordata, 3 different branches. And I'm not going to, I'll let these branches be tritomine. Although they are resolvable, I'm not going, for simplicity, I'm just going to put them together like this. Okay, one of these is something called cephalochordata. You'll see some of these in the lab. Actually, let's put it over here. Okay, cephalochordata. These things are marine. You usually find them on beaches. They bury themselves in sand or gravel with only the head sticking up, and they're very segmented. Okay, the muscles there are in segments, and this segmental muscle actually is maintained pretty far up in the phylogeny. If you eat fish, you can see the segmental muscles. They're arranged, you know, in layers like that. And so the segmentation is still there. These things are called lancets, common name. They're filter feeders. So here they take in food into, or water that is organic rich, into the pharynx. And once again, the pharyngeal slits here function as filter feeding. And so also a little bit for gas exchange. Then that comes through the slits and the water then escapes out another piece of the body. Other than that, I'm not going to say any more about them. I'm not giving any apomorphies for these groups, okay? Another chordate group that you don't come across very often, is not very abundant, is Earl chordata. And the adults look more like plants. Okay, they don't have hardly any of the chordate apomorphies that I mentioned before, except, however, the larvae, once they hatch out of the egg, the larvae will swim around, and they have a dorsal nerve cord, hollow nerve cord. They have a notochord. They have a post-anal tail. They have all the features that you associate with a chordata. However, when they settle down, they will metamorphose into something that looks more like a coral. And these are called tunicates or sea squirts. They're usually very small animals living in colonies, and they are filter feeders, okay? They've lost all of the chordate features in the adult. So these are sessile. Sessile meaning they stay in one place, they don't move around. And so they take in water, once again, into the pharynx, and the pharynx is really huge. And it filters out food particles. So as the water will move through these slits into an area around the animal, and then out a siphon-like opening. So the water is coming in, going through, and out. Okay? And so this group does have one apomorphy that I'll give to only to this group, is that it secretes a layer almost like a cuticle. Okay, however, it is of a different origin. It consists of collagen protein, and it's called a tunic. So that's on the outside of the epidermis. Okay, so we won't worry about tunicates anymore. The rest is the largest group, Craniata. Craniates, it's a group we belong to, and at the anterior end of the nerve cord is is a cluster of ganglia, nerve cells that constitute the brain. And this brain is within what is called cranium. Now, this is not made of bone. Don't mistake cranium and skull. They're not the same thing. Okay, they're later, but a cranium is usually made of cartilage. And we, as embryos, we do start with cranium. Eventually, dermal bone develops, and it replaces or incorporates the cranium. And so usually we don't see the cranium except for in embryos, but in these groups, lower groups, that is pretty significant. Okay, and this is a photograph of some of the earliest vertebrate fossils or craniates. This here, fish-like thing, segmented muscles. It was also a filter feeder, has a name, but we're not going to worry about it, okay? This occurs back in the Paleozoic, this particular fossil, the early Paleozoic. One group here, I have the scientific name, but we're going to you can just call them hagfish. Okay, there's about 3 or 4 species of these things, and they're morphologically pretty primitive, eel-like. They do not have a vertebral column, but they do have cranium around the brain. Okay. Furthermore, they have an open circulatory system, so they don't have blood that's in vessels. Okay. They do have a heart, and the heart just pumps the fluid around the side of the body, but it's an open circulatory system. You don't need to know them as Vixini. You can just call them hagfish. They're all marine. They're all marine. And when they're disturbed, they will secrete abundant amounts of mucus that with their predators get tangled up in. The rest of the craniates, vertebrata. Okay, so craniates, vertebrates, not the same thing. Okay, craniates includes hagfish, but vertebrata is everything else. Okay, here you have the nerve cord is in a protected sheath that is segmented, called the vertebrae or backbone. Well, let's not call it a backbone because at this stage it's just cartilage. Okay, there's no mineralization to it, but still you have box-like structures that go around the nerve cord, and they have areas of flexion between them so that you can move. So this is the vertebral column, and that is the only apomorphy I'm going to give for vertebrata. And this is usually the group that we refer to as vertebrates. Additional things here with the vertebrates is by this time, in the hagfish, although you can tell when it moves where its head is, it doesn't have too many features there for identifying the head and differentiating the head from the rest of the body. Whereas in most vertebrates, it's pretty easy to see the head. Okay, it's pretty developed. That's what I mean by cellularization. Okay, this group can also give you a fancy name, but you can just call them by the common name, lampreys. They look like hagfish. They superficially, they do look like hagfish. However, they have the internal morphology is more developed. They do have vertebral column, around the backbone. There's another group. In fact, these are the only survivors of a large group of jawless fish that existed down here during the early Paleozoic. All of these were, the Paleozoic ones, they were probably 8 debris off of the bottom, okay, of the ocean. They didn't, they weren't predators, they just fed on detritus. Whereas the modern lampreys are all happen to all be predators, or not predators, but parasites. They suck the blood of other animals, especially fish. So they don't have jaws, they don't really have any fins, sort of eel-like, and they have these, inside their mouth, all these teeth that cover the whole inside of the mouth, and sucker-like organ, and they latch onto the side of a fish, and they will, rasping, okay, I'll put this up there, rasping mouth parts, in which they cut through the skin and then suck blood. Okay, these are usually on larger fish, like shown right there. Okay, it looks like this is probably all the further I can go, except I just want to, they only have dorsal fins. This is the dorsal fin right there, but nothing on the sides. And skeleton, just cartilage. And their heart is more sophisticated than in these earlier groups down here, and that it consists of 2 chambers. Okay, and I'll come back to this on Thursday and describe what I mean by that. Okay, this is where I will stop.